Thank you, Professor Wang. Please stay up here, and I would like to call Professor Craig Venter too. So now there will be possibilities for the young people to ask some questions. I have, as a matter of fact, one question already here, and and I don't know who of you should be the best to answer that. So the question I have is that we heard from both of you about the partners we have in the guts, the microbes. And my question is, from one of the young people here, are they important for such things as your temper, your psychological behavior? What do you think?
Actually, my talk is even no more than half of Craig Venter's talk. He talked about both reading and writing genomes. I just talked about reading genomes. For this one, we have read some of the genome sequences of the bacteria as a part of what we call the meta-genomes in our gut. We realized important. First is that classical experiment, they change just everything in the gut between two mice. One is genetically fat, the other is genetically thin. Just by changing the bacteria in the mice's guts, and then the phenotypes or obesity has been totally transferred from this one to another one. We also have done so. And we are working very hard to find what bacteria would be responsible. I can tell you also, on mice, we do have found dozens of bacteria which play very important roles, not only to obesity, but also to diabetes. So especially, another example might be the Chinese medicine. Perhaps the possible effect of Chinese medicine dependent very much, to some extent, to those bacteria in our guts. Then, of course, as Craig Venter mentioned today, at the very beginning, after being at birth, we did not have any bacteria in our gut. And then we also have found the major species of bacteria actually are inherited. Or demonstrates an obvious family character. And then, of course, it could be interpreted by non-genetic reasons. But as a conclusion, I do think that the bacteria in our bodies, which now we call other genomes of human beings, would play important role in our daily life related to our health and diseases. Thank you.
So your brief answer is yes.
My briefer answer is we don't know yet, but I agree they must have a role. So if you measure how many chemicals are in your bloodstream right now, particularly after a meal, there's about 500 chemicals circulating through your bloodstream. 10% of those are from bacterial metabolites of human chemicals, and the other 30% or so that you absorb from food in your diet. So that means 50 chemicals circulating through your brains right now are from bacterial metabolites. So the microbiome is a huge part of our metabolic potential. We have no idea what those 50 chemicals do, whether they make you happy, sad, sick, successful, you know. I want to know what he's been eating.
No, we all know.
So may I ask, excuse me, may I ask the young people who have questions to raise a hand? We have microphones in.
And then you will receive that invaluable reward or gift, his book, one of his books.
In the meantime, I could ask you, Craig, so if it is in that way, then of course, from Darwinian point of view, we must have a control system so we could select and control the...
Yeah, your microbiome is determined as much by your genome as it is by the environment. In fact, early on, with the tremendous role of H. pylori being associated with stomach ulcers and stomach cancer, I think it's similar to what Henry said. You can actually track relationships of people and families totally by the H. pylori in your gut. So, you can find out who your girlfriend is and your wife at the same time from H. pylori.
So please.
Yes, that's right.
This is a question not particularly on the current topic, but what is your opinion with the increasing rate at which biological science is advancing? So do you think, how drastically do you think human society will be changed by these advances in biology? Do you think transhumanism will be a possibility in, say, 20, 30 years' time? Just how drastic would this extent of transhumanism be?
So that's a challenging question.
I mean, it depends on how we use this information. What you can see is the new technology in genomics has caused, and we just heard it, a huge explosion in our ability to generate information. The challenge for you and all the young scientists is how do you convert that information into knowledge? And the pace that happens will determine the future. If we can add knowledge at the same rate that we're adding base pairs of data, it's going to be a very exciting future, and anything is possible. But there's also political implications. How do you use the data? How do you use identification data? How do you use the fact that we haven't talked about yet, that we're probably much more genetic species than we like to think we are? All those are going to become important as we draw the lines between diagnostic and predicting medical futures to making society selections on people based on their genome.
More questions? Yes? You could go to the microphone, yes. I have on the list here, and I would like to ask you, if you were not a scientist, what would you be?
No, first, it's very clear, still a human being. Scientist, to be a scientist has never been my unique choice. But the reason for me to have chosen it is to learn from him and other pioneers. We can imagine, without sense, what life would we still have been living. In this respect, that's also a partial answer for the previous question. Life in nature is ethical. If possible, if allowed, I just hope all of the people would become scientists, even though I'm not highly qualified.
Very good. I almost misread it. If you were not a human being, what would you be?
If I'm not a human being, I would be, anyway, something with a genome of DNA or RNA.
Please.
Yeah, I'm actually not that young. I'm 33, but you all have made me be young again, and I remembered my childhood ambition was to be a scientist. So prior to this, I was actually a teacher for 6 years. So this year, I just started my PhD because I thought it's about time that I realized my dream of becoming a scientist. So I have to thank you all to make me a child again. I'm young at heart, but I know I'm quite old. So these are some of the questions I have. I mean, it's a great opportunity to learn from great scientists like you all. So I have a few questions. So I hope there are not too many. Okay, like for example, Dr. Craig, okay, I understand that you went for a tour of duty as a Navy corpsman in Vietnam. So I want to understand, I mean, I like people, I mean, I want to know more about people. So like for your case, you went to tour of duty in Vietnam. How did it shape you as a scientist?
That's very personal.
That's very good.
And that's the first question.
If you win my book, then you'll have the answer. But
How much is the book?
no, it shaped me a lot because, in fact, in contrast to all of you, I was a very poor student. I didn't like school. I liked building things. I liked playing sports. I liked having lots of girlfriends. And so I almost did not graduate from high school. I came very close to not graduating, but I moved to Southern California to take up a surfing career. And I got drafted and sent to Vietnam, and it totally changed my life because I worked in medicine and I found that knowledge was power. And the more I knew, the more lives I could save. And it was the first real introduction in my life. It was a difficult situation to learn it in, but it was a very powerful lesson. So my plan was to go to medical school because I really liked practicing medicine. But when I went to the university, I was introduced to high-end science. And it was the perfect outlet for my curiosity that was not destroyed by the education system. So maybe I've been successful because I ignored the education system. I went outside, and I still had my curiosity. And I made an early breakthrough in science, proving adrenaline worked on the outside of cells as an undergraduate, and published my first paper. And I was so hooked that you could take ideas, ask them about life, do careful research studies, and prove those ideas. And I became so hooked on it, science became my career.
Just second him by saying 2 points. First, I don't know it's fortunate or unfortunate. There are so many young staffs at the BGI who don't want to graduate from the high middle school, not a university, even not a PhD. They don't like school at all. Then the second, I have not experienced any war, but one of my most important hobby is to visit all of the battlefields. I always think, no more, it's too expensive. And then also contribute to all the death with you, with your experience. We will never have war again. Unfortunately, there are still some war even locally in this world. War, really impressive at least. So congratulate that you have that special experience.
Okay, my next question. Okay, the first human genome was actually sequenced, which was actually your own genome, right? So where did you harvest the cells for this, and why your own genome? Why not someone else, probably the president of USA? You know, why you chose your own genome, and which part, you know, of your body the cells came from? I mean, if it's in the book, you don't have to answer, I'll buy the book.
So in the early parts of genomics, geneticists were very afraid of genomics. And the big fear was if somebody's genome was sequenced and published, that there would be severe repercussions against those people. They would not be able to get health insurance. There might be other, they might not be able to keep their jobs. There was tremendous fear. It was a fear-driven program coming out of traditional human genetics. And so the idea of informed consent, if I ask you and say, I have to convince you you should have your genome sequenced, and it's really okay, there's no negative consequences. Maybe some of those consequences were real. Who better has informed consent? I knew what I was doing, and it's easier to ask yourself than to ask somebody else to take that risk. In the end, there ended up being no risk, and we've changed the paradigm because now it became the standard to publish your genome on the internet instead of something that was forbidden by bioethics committees. So sometimes you just have to take the risk. So, you know, it was not an act of ego that I thought I was the prototypical human that everybody should be modeled against. Oh, that's not a bad idea. But it was about the risk and the risk involved in the perception of those risks. And I think Barry Marshall proved this very nicely with the discovery that H. pylori was the cause of stomach ulcers. He could not convince anybody scientifically, so he swallowed the H. pylori himself to prove that in fact it was the cause. It's not an experiment you can do to somebody else. So self-human experimentation has been a key part of medical advances throughout medical history.
Again, from my part, it reminds me of a Chinese saying, 陆地即实地. When you are worried about what would happen, all terrible things after falling down, it's better for you to fall down to the ground. And then you will find actually it's not that risky. Of course, it's not for every case. But for me, I still think the first generation or the group of people who have their genome sequenced are pioneers, are contributors. I would say they have not benefited so much from the genome sequence as you. That's a contributor. Then I have proposed, it raises a serious ethical issue. That's the ownership of your genome sequence. In the past, based on that individual informed consent, we will just sign that informed consent form that I'm going to contribute my genome. But now it's really a problem from the long-term point of view. What about my son? Someday, if he would or were unhappy, he would sue me. How can you release part of my genome? And then his mother, my genome minus half of my son's genome is half of her genome. That's the reason I call for anonymization, not to release the name and the medical information related to name together with the genome sequences. I would ask those who would like to be sequenced, please raise your hands. Very good. Thank you very much. We understand each other.
Because you had so good questions, you may have one more.
Oh, one more. Okay.
But before you ask that, I have a more frightening prospect
one question
for you.
probably.
So, because my genome was the first one sequenced and it was on the internet and it's been broadcast into space now, the frightening prospect is we might get visited by 10,000 Craig Venters in the future.
Yes, definitely. That's the first sequence, the man, the genome, that's right. Yes, please.
Okay, this question is posed to both Prof. Yuan Ming and also Huan Ming and Dr. Craig. Okay, do you envision a future in which job interviews no longer exist and companies make their selections based on their genomes? Or if you have watched a blockbuster movie, Get a Car, in the 90s, early 2000s, do you think that is where we are heading? That means I don't need to go for any job interview. You just take some of my cheek cells and then you just tell me whether I'm
Would you
I'm hired
please
or not.
repeat this question again?
Okay, do you foresee a future whereby human beings do not need to go for job interviews? Rather, you would just harvest our cheek cells and then you look at our genome and then you decide whether you want to select us as your employees. Do you envision that future?
You may not know, but that's how you were selected to be part of this audience.
I thought so too, yeah.
We sequenced your genome from the fingerprint on your application.
Okay, one last question. This one I really need to know. Okay, Osama bin Laden, right? Just now you were showing a lot of dead bodies. So is Osama bin Laden been verified as Osama bin Laden? Because you showed that Saddam Hussein is Saddam Hussein. So I'm wondering, is Osama bin Laden really Osama bin Laden? Have the American president got in touch with BGI?
I think that's for you.
So dear, the previous question, it sounds like very much that Hollywood movie, Gattaca,
Yeah,
or we
Gattaca.
pronounced it as Gattaca.
Yeah.
At that time, it is true, in a second, your genome will be sequenced with the sample which has been taken from you without any attention of yourself. And then now, actually, it is possible because every single cell falling from your fingerprint would contain 6 picograms of DNA. with all the actual kits provided by the companies, if you put them together, you really can have enough amount of DNA for sequencing. I think the point is whether sequencing or sequences will be used for the benefit of the future or not. I'm very confident. I was taught at my childhood that the 21st century would be the century of biology. I think now we can hear steps closer and closer to us.
Let me give you a more serious answer to your question. So I think one of the fears that came out of human genetics, human geneticists were studying very rare diseases, the so-called monogenic diseases, where a change in a single gene could be linked to a disease. They were studying those by definition because they were rare diseases. Most things that happen to us in our physiologies do not fall into those categories. And so if you extrapolate from these rare single gene disorders, everybody thought early on that every part of human existence would be explained by a change in one gene. And that's why the biggest surprise in 2000 when we announced the completion of the genome was that we had only 20,000 something genes, because people were predicting we would have 200,000 to 300,000 genes because we had to have a lot to explain all these different characteristics if it was one gene per characteristic. It's a frighteningly naive approach that's come out of modern science a decade ago even, that's what the thinking was. To interpret your genome, to interpret my genome, you have to understand probability statistics. There's very few yes-no answers in any of our genomes. There's probabilities of things happening. And that's why we need so many genomes with well-characterized phenotypes to narrow down what those probabilities are so we can give each person meaningful information. So it can never be used intelligently to select you for a job or a career or being an athlete.
Sure, agree.
Well,
Okay, thank you very much.
Thank you. We should try to wrap up the discussion. I see that we have in the audience. Okay, one more question.
Mr. Craig is so gentleman.
Thank you for giving me this chance. My name is Ning. I'm currently a third-year bioengineering student in NTU, and I'm from China. I have two small questions for Dr. Winter. First of all, currently I'm doing a research attachment at Institute of Bioengineering and Nanotechnology, where Prof. Jackie Ying is the doctor. And I'm wondering, the IBN has regular open houses for the public, also like students from tertiary universities and JCs. I'm wondering if the nice building of Venter Institute in San Diego have such open houses for the public.
Yes, we do, and we have visitors all the time. In fact, we have a fellowship internship program where everything from high school students, college students, medical students, graduate students can come and do internships in either Rockville or San Diego. And we have high numbers of visitors all the time. So you should check online at jcvi.org and you can come do a fellowship there.
Thank you.
I'm very proud of you. I would be very happy if you say it to me. No, that's true, that we have totally open system. Many students who have been working at BGI for months, but still, we don't know them. You are most welcome, every of you, to come to BGI.
Yes, I also come from my
he
friend.
told me earlier he hates yellow sweaters. We like yellow sweaters in California.
Thank you.
That's a royal color, emperor color.
Thank you very much.
I think with these words we end the questions and answers.
Sorry, one last question.
You have one more?
Yeah, the last question is more technology oriented. I'm very interested in computational biology, and I think using computer software to solve synthetic biology problem is very promising. And my question is like, what's the function of the synthetic organism designer, which is developed by the Venter? Sorry. Is it function as a simulation of the result of the synthesis. But considering the complexity of the protein encoding system and the metabolic network in the organism, it sounds very challenging to just use a software to simulate it.
Who can give the shortest
Now,
answer?
it's actually an excellent question because you're pointing to what has to be done for the future. We don't have computer simulations to model life right now, in part because we don't know all the first principles of life. We have all these unknown genes that are essential functions, but we don't know those functions yet. So it's impossible to model them in the computer. But if we can build by first principles like we're trying to do a minimal cell, and that lives, then we can work back and start to build models in the computer to do this. And it's a matter of getting enough data, enough iterations, understanding that we're still in this empirical phase of biology. And until we make enough discoveries, you can't do the proper computer modeling, but it can be done on pathways now, and it can be done trying to put it together. So I think it's going to be one of the most important advances in the future. And we're helping, Ham Smith and I are helping the bioengineering department at the University of California, San Diego, start a genome design study program to try and do just that, but it's in the very, very beginning stages. So there's lots of rooms for major discoveries when you come to visit us, you can work on.
Thank you, both
Thank
of
you.
you. And with that, we close this session. So thanks.